We're trying to understand the genetic pathways for diseases like cancer to help improve diagnostic tools and treatments. My research is focused on understanding the regulation of growth control during development and diseases like cancer. My work is trying to answer questions like, why do cancers grow so quickly and become so big? Understanding genetic pathways behind growth can help answer questions and investigate how cancers can be so detrimental. We use the fruit fly or Drosophila melanogaster to model cancers like gliomas. Gliomas are primary brain tumors that progress into glioblastoma multiforme, an aggressive and fatal brain cancer. Interestingly, these tiny flies have the same cancer-causing genes and machinery as humans. With that, our goal is to study the early changes that happen in cells which change them from normal cells to cancerous cells. Since fruit flies have a vast array of molecular tools, using them to study early genetic and molecular changes is ideal. My research identifies the minimum number of genetic changes that transform normal cells to cancers and find evolutionarily conserved pathways that drive cancer growth. Towards this effort, I was part of a group that identified the Hippo pathway, a major growth regulatory pathway now implicated in promoting cancer growth by its interactions with other cellular pathways. In my lab, we identify molecular networks that interact with the Hippo pathway for cancer growth. This work has a larger impact on patients since genome analyses and oncotypes are major diagnostic tools physicians use to identify cancers and provide appropriate treatment. Our long-term goal is to find key drivers of cancers like glioma to design better inhibitors of these pathways for more targeted treatments. A second area of interest is to investigate molecular mechanisms underlying neurodegeneration, specifically the role of Hippo signaling as it controls both cell division and cell death. Neurodegenerative disorders like Alzheimer's disease are so called because of the progressively increased loss of neurons due to cell death caused by accumulation of amyloid beta or tau proteins. We have developed genetic models of amyloid beta 42 and tau-mediated neurodegeneration in Drosophila melanogaster. Using these models, we found that the Hippo pathway affects amyloid beta 42 and tau-mediated neurodegeneration. Using funding from the STEM Catalyst grant, we will study the role of Hippo signaling in neurodegeneration. For these studies, I collaborate with Dr. Amit Singh from the Department of Biology at the University of Dayton. Our goal is to test if inhibiting Hippo pathway suppresses neurodegeneration using our fly models. Another collaborator, Dr. Uday Pandey, who is at the University of Pittsburgh Medical Center, will test if findings from flies are relevant to mammalian models of AD. These basic studies will help understand the pathobiology of Alzheimer's disease and related dementia. Our studies will identify candidates that could be tested for their diagnostic or therapeutic value.